Well, it looks like we've got a quorum, or do you want to wait till you hit 50?
I think we're in pretty good shape whenever we want to get started here.
Great. Okay, well, welcome to all of you for showing up, virtually anyway, for this final stage in Jared Beck's PhD career. I woke this morning, like many of you, to a dawn chorus of birdsong heralding the spring. Birds are busily building nests to fill with eggs that will soon hatch to become greedy chicks, which will gobble up all the food that parents bring them until they can leave their nest to launch their own independent lives. Jared, a native son of Wisconsin, has always liked birds. He considered studying them as a student at Carleton, but he had his head turned there by the charms of plants and prairies as he absorbed the ecological knowledge generously shared by his mentor, Mark McCone. Upon graduating from Carleton, he continued working as Mark's research assistant while beginning work as an intern with Stuart Genius at the Chicago Botanical Garden. He thus arrived at the University of Wisconsin with a lot of experience and skills, simplifying my job. Thank you, Mark. Today, and Stuart, today we'll hear about the research that's filled so much of Jared's life these last 4 years. We also assemble to help Jared celebrate as he fledges from the nest of UW's botany department. Ecology is broad as a science, ironically full of specialists. Jared is not one of them. I like that about him. Jared is also smart, informed, highly motivated, well organized, and he plans ahead. I learned that he plans ahead when he wrote to me more than 7 years ago to say, I've recently developed a strong interest in the ecological population and genetic consequences of habitat fragmentation for plant communities. My work in the Carleton Arboretum has led me to wonder how fragmentation shapes the ecology and genetic structure of isolated populations. I encouraged him, of course, he looked good, and I was pleased when he finally decided to come to UW 3 years later, NSF fellowship in hand, to work on a fascinating and diverse set of projects that you'll hear about shortly. None of these, however, concern the genetic structure of isolated populations. Sorry, Jared. Instead, you'll learn about the ecological processes Jared pursued. These extend over fine to coarse spatial scales over short to long spans of time, over many field sites to inside computers, and from field observations to clever experiments. You'll learn something about the particular characteristics that affect how forest herbs grow and thrive in environments that differ greatly in climate, light, soil texture, and nutrients. You will see how Jared applies cutting statistical models to understand how plant functional traits affect their responses to environmental gradients. You will see how Jared analyzed the fine-scale spatial distribution of species to learn more about the social lives of plants and their ability to partition and share niche space. When Brett Largett and I were trying to address a macroecology problem, Jared jumped in to help. The problem was this: how do we infer rates of colonization and extinction over many sites, given that all samples are incomplete and thus miss some of the species present? Solving this phantom species problem was technically challenging. We took full advantage of Jared's R skills. The work represents an important advance, but you'll not hear anything about that today because Jared elected not to include this work in his thesis. You will also not hear about how fire synchronizes flowering and boosts reproduction in a widespread but declining prairie species. The title of his work on Echinacea angustifolia, published with Stuart Wigenius and Gretel Kiefer in PNAS this past spring. You will, however, learn how Jared moved beyond observational studies to perform experiments to rigorously test the extent to which seedlings are helped or hurt by local microbial friends and enemies that surround them in the soil. Most broadly, you'll see how Jared is working to assemble various ideas and approaches to improve our understanding of how plant species respond to gradients and conditions and assemble into plant communities. Jared brings many attributes to his work. First and foremost, he's motivated by his deep concern for the natural world and lively scientific curiosity. He also brings great depth and remarkable balance to his science by combining his interest in natural history with interest in theory, by working alone and with others, by being a great student and a gifted teacher, and by wielding a chainsaw and R code with equal comfort. These functional traits served him well as a graduate student, but what will he become? That's difficult to predict given his biomarkers. He's a social loner, a birding botanist, an informed conservationist, and a promising quantitative ecologist. While many career paths lie open to him, I cannot predict which he will choose. All of us, Jared's academic parents, siblings, aunts, uncles, and cousins, as well as his actual family, share pride in Jared's academic accomplishments and wish him well as he disperses to pursue the next stage in his career. I invite you all to join us after his defense for a specially catered reception in the Botany Garden. Wait, I've got a notice here. My God, there's an appendix. It's forcing a last-minute change in plans. Instead of that shared reception, apparently you're being asked instead to don a face mask, make your way outside on a pretty day, and celebrate spring and Jared's success and his sometimes socially isolated personality with a toast and a suitable distance, of course. Be sure to also send him your greetings and well wishes by an electronic means of your choice. If you happen to be able to offer him a remunerative and prestigious job, ideally in the Baraboo Hills, I'm sure he'd be happy about that too. Let's now listen to Jared Beck's PhD talk, Factors Shaping the Distribution, Abundance, and Diversity of Temperate Forest Plants. Jared?
Thank you, Don. Can everyone hear me? Let's just make sure we have that working for us right now. Excellent, thank you. All right, thank you, Don, for that kind introduction, and thank you all for attending my virtual exit seminar. While many of us are now spending our days cooped up indoors, I hope you've had an opportunity to get outside and enjoy some fresh air. Perhaps even find some spring wildflowers in bloom. For those of us in the temperate regions of North America, the arrival of spring is accompanied by a spectacular display of spring wildflowers in many of our forests. With the incredible variation in flower color, shape, and size, even those without advanced botanical training can recognize and appreciate the remarkable diversity of herbaceous plants found in temperate forests. In these forests, herbaceous or non-woody taxa comprise 80% of all plant species, including these charismatic spring wildflowers, as well as many less showy species. Between 50 and 100 herb species can coexist within 1 hectare of forest, approximately the size of 2 football fields, and you may find 10 to 20 species co-occurring in a single square meter. Given that these species compete for the same basic resources, such as light, water, soil nutrients, and others, what factors maintain the remarkable diversity of forest herbs? In my dissertation, I seek to address 2 fundamental questions about the diversity of temperate forest herbs. First, what factors influence herb distributions across the landscape and within forest stands? And second, what mechanisms maintain herbaceous plant diversity? To address these questions, I studied temperate forest plant communities in Wisconsin. Here, an exceptional legacy of research in plant ecology and the existence of exceptional data sets facilitate analyses of temperate forest herbs. My research includes broad-scale studies of plant distributions across the state of Wisconsin, as well as more focused studies of plant distributions within the Baraboo Hills. This area, just an hour north of Madison, comprises the largest intact forest ecosystem in southern Wisconsin and is a regional hotspot of plant biodiversity, supporting more than 1,000 plant species. There are a variety of ecological processes that can influence community assembly, so I find it helpful to reference a conceptual model that is currently popular among ecologists. In this conceptual model, community assembly can be broken down into a series of hierarchical steps that affect 3 distinct spatial scales. Starting from the left and moving to the right, we start with the regional species pool, which in the context of my research includes all plant species found in the state of Wisconsin. We can then understand how plant species go from the regional species pool and sort into local communities, which are groups of populations occupying the same geographic space. In the context of my study, these are relatively homogeneous forest stands. And finally, we can ask how plants are distributed within local communities among microsites, which constitute the area in which plants are directly competing for resources. Recently, ecologists have begun to emphasize that different ecological processes may structure communities at different spatial scales. In other words, the processes influencing how species sort from a regional species pool into local communities may differ from the processes shaping how plants are distributed at more local scales. In my talk today, I will examine the factors that influence herb distributions across local communities, as well as the distribution within local communities. This approach will allow me to test whether common predictions made by ecologists about community assembly theory are consistent with empirical data from temperate forest herbs. Today, I will start broad and ask what factors plant distributions across the landscape. Remember that in the context of my study, the regional species pool includes all herbaceous plant species in the state of Wisconsin, and those local communities constitute forest stands with relatively homogeneous environmental conditions. Understanding the factors that influence plant distributions across the landscape has a long history in ecology. Over 100 years of research on this topic have informed current predictions about what factors influence broad-scale plant distributions. In fact, work in temperate forests conducted by John Curtis and his students here in UW-Madison's botany department more than 60 years ago laid the foundation for our understanding of how environmental variation shapes plant distributions and ecological communities. In the time since Curtis, advances in physiological ecology suggest species responses to the environment often depend on morphological or physiological characteristics, sometimes referred to as functional traits. Modern community assembly theory predicts that species distributions across the landscape result from these trait-environment associations that reflect different strategies for acquiring resources that are advantageous in different environments. In this study, my goal was to evaluate whether plant distributions across the landscape reflect environmental variation and whether species-environment relationships could be predicted by functional traits. To investigate how these trait-environment associations might shape plant distributions across the landscape, I analyzed an existing dataset describing the distribution of plant species across the state of Wisconsin. These data were collected by past students in the Waller Lab between 2000 and 2012. And these past students resurveyed sites that were originally surveyed by Curtis and his students. My analysis focused on 259 forest stands, shown in this map here, and 139 herbaceous plant species for which we had functional trait data and were sufficiently abundant. To analyze these distributions across the landscape, we formulated phylogenetic multi-level models, which are cutting-edge statistical models that help analyze how the probability of finding a species within a given site varies with environmental conditions, the traits of those species, as well as their phylogenetic relationships. Our model included 4 environmental predictors, including soil sand, soil calcium content, average tree basal area within those sites, and the ratio of precipitation to a potential evapotranspiration, which reflects how much water is actually available to plants, as well as 4 functional traits that we have data on for each species, including vegetative leaf height, specific leaf area, leaf carbon to nitrogen ratios, and seed mass. At its core, the statistical model characterizes plant distributions in relation to environmental variables. For example, in these 2 graphs on the right, you can see how the probability of finding Michella repens at top and Sanguinaria canadensis varies with the amount of soil sand present in a given forest. Using these models, we can then quantify the extent to which these species-environment relationships depend on functional traits or phylogenetic relationships. Today, I will be just talking about the influence of functional traits on those species-environment distributions. Just as John Curtis and his students demonstrated more than 60 years ago, we found that plant distributions varied along environmental gradients in Wisconsin. But we can go a step further to examine how these species-environment relationships depend on functional traits. And rather than show you all combinations of the 4 environmental variables and 4 traits that could influence these distributions, today I just want to focus on plant responses to soil sand. And in the top graph, I'm going to show you estimates from our statistical model. Estimates that are positive or fall to the right of that dashed line indicate that there's a positive relationship between a particular trait and the probability of finding, and the relationship between the probability of finding a species and increases in soil sand. This will become a little bit more clear in the bottom 2 graphs as we go along here. So when you look at these point estimates, the takeaway message is that leaf height and specific leaf area do not mediate how plants respond to changes in soil sand across the landscape. However, both leaf carbon to nitrogen ratios and seed mass are associated with plant responses to soil sand. For example, plant species with higher than average leaf carbon to nitrogen ratios increase slightly with soil sand, but the probability of finding species with lower than average leaf carbon to nitrogen ratios declines rapidly with soil sand. This may reflect the competitive advantage of having high carbon to nitrogen ratios in environments with reduced nutrient availability and increased water stress, such as sandy environments. In a similar way, we can look at seed mass, and we find that species with greater than average seed mass decline with soil sand, while species with lower than average seed mass decline only slightly with changes in soil sand. This may reflect the sparse herb coverage and reduced competition for light in sandy sites, and whereas in contrast, large seeds may be advantageous in environments where light is more limiting. Our investigation of plant distributions across the landscape revealed strong trait-environment associations that are consistent with expectations that environmental sorting plays an important role in community assembly. Specifically, plants with traits that allow them to physiologically tolerate local abiotic conditions and compete successfully within given environmental conditions lead to strong environmental sorting that's mediated by functional traits. These results are consistent with predictions from modern community assembly theory. Now that we have a handle for what factors are shaping plant distributions across the landscape, I want to turn my attention to what's happening at finer spatial scales. Specifically, what factors influence local plant distributions and maintain herbaceous plant diversity within those local forest communities. Many ecologists predict that resource competition is the most important factor structuring local communities. And more specifically, when we have species that are able to coexist with one another, the common assumption is that they are using resources in a complementary fashion. If such complementary resource use structures herbaceous plant communities, we expect that co-occurring species should exhibit niche differences associated with these complementary strategies for acquiring and using resources. In other words, we might expect that functionally similar species will be less likely to co-occur because they compete more strongly for the same resources. Using similar logic, we might expect that phylogenetically related species would be less likely to co-occur. If close relatives share ecological characteristics as a result of their evolutionary history, we would expect those species to compete more strongly with one another, leading to competitive exclusion. To evaluate these predictions with empirical data, we use the same data set consisting of 139 herbaceous plant species across 259 forest stands. But this time we looked at plant distributions and co-occurrence patterns within each forest stand. So we examined patterns of co-occurrence within the 1 by 1 meter quadrats in which plant species were surveyed. And notably, this is the scale at which plants are directly competing for resources. So if there is complementary resource use, this is the spatial scale we would expect to see it. Within each forest stand, we modeled plant co-occurrence patterns within quadrats and fit models with specialized covariance matrices that specify that functionally similar or phylogenetically related species were less likely to co-occur. We could then compare these statistical models that include a term that specifies that functionally similar or phylogenetically related species co-occur with models that don't include that term to understand how much that improves the model fit. And so we quantified the model fit using this deviance information criterion, or DIC. And the key to interpreting these graphs is this key value of a DIC of 5. So I'm going to show you a histogram of the difference in performance scores across 259 forest stands. And the way you can interpret that histogram is sites that fall to the right of that dashed line. So when the change in this information score is greater than 5, there's strong evidence that either functionally similar species segregate or that phylogenetically related species are less likely to co-occur. Starting with functional similarities, surprisingly, we found little evidence of niche differentiation among co-occurring species. Just 12 of those 259 sites showed evidence that functionally similar species were segregated from one another. Similarly, just 13 sites exhibited phylogenetic repulsion. These results suggest there's little evidence that co-occurring herb species in temperate forests exhibit niche differentiation and complementary resource use. While many ecologists predict that coexisting species should exhibit complementary resource use, we found little evidence that co-occurring forest herbs exhibited such niche differentiation. Although our data was only a snapshot and could obscure important dynamics that play out over time, these results are inconsistent with common assumptions about the role of niche differentiation in structuring local communities. So the question I started with remains: what factors promote local coexistence among forest herbs? I identify 3 possibilities that I'm going to spend the rest of my talk discussing. The first are equalizing processes, which I think are best explained in the context of environmental sorting. When species sort into different environments, species that are mismatched to a given environment are often at a competitive disadvantage and will likely be excluded. So in this little conceptual diagram, we have 2 sites varying in soil characteristics, the gray site and the brown site. And what we can see is that environmental conditions in the brown site are favoring the taller species, and the shorter statured species is competitively excluded. Whereas in the gray environment, perhaps that large statured species is unable to survive or tolerate local conditions, and that favors the smaller statured herb. When we have strong environmental sorting, species that co-occur within a given environment often exhibit many ecological similarities. And while these co-occurring species are not ecologically equivalent, and there could be important competitive differences, these competitive differences are relatively small. So competitive exclusion will happen slowly relative to local dispersal and other factors that influence local plant communities that would scramble up the community before competitive exclusion can happen. And so these equalizing processes could promote coexistence among temperate forest herbs at local scales, or at the very least set the stage for other processes, comparatively weaker processes, to operate. Second, just as environmental variation across the landscape influences plant distributions across Wisconsin, as we saw, fine-scale environmental variation within communities, but at scales larger than those microsites, could influence local plant distributions and lead them to spatially partitioned microsites within forest stands. Finally, interactions between plants and natural enemies, such as pathogens or herbivores, could promote coexistence by preventing any one herb species from becoming too abundant. The remainder of my talk will evaluate these 3 potential factors influencing herbaceous plant communities. To start, let's take a closer look at how equalizing processes might influence plant distributions and the potential for local coexistence. Recall that strong environmental sorting and little evidence of niche differentiation at local scales suggest that equalizing processes could influence local herb communities in Wisconsin forests. To illustrate how these equalizing processes might operate, I want to focus on two closely related herbs. Here we have the two species growing side by side in the forest. On the left is Hylodesmum nudiflorum, or the naked-stemmed tick trefoil, and on the right we have Hylodesmum glutinosum, or the pointed-leaved tick trefoil. If you're not familiar with those names, perhaps you remember their old name before taxonomists kind of played a little shell game with us, and that old name is Desmodium. And if you aren't familiar with those names, you may be familiar with their mode of seed dispersal. If you've ever taken a walk through the woods in late summer or early fall and brushed up against the seed pods of Desmodium, you'll find that it has tiny Velcro-like hairs that attach to your pants, and you can spend hours pulling them off. Or perhaps you might have annoyed your coworkers while surveying prairie vegetation as an undergraduate, and your dear friends gathered a softball-sized glob of Desmodium seeds and smeared them down your back. Despite my own traumatic experience with Desmodium, I found this pair of species fascinating. During my fieldwork, I became intrigued by how frequently these species occurred side by side. As an ecologist, their widespread co-occurrence posed a coexistence conundrum. Close relatives often share ecological characteristics as a result of their shared evolutionary history, and these trait similarities should favor co-occurrence in suitable environments. Yet species that are too ecologically similar are expected to compete more strongly with one another, leading to competitive exclusion. Thus, when we find close relatives that are coexisting with one another, the common expectation is there's some sort of niche difference that allows these species to coexist. In this study, I wanted to better understand what factors promoted their widespread co-occurrence. And my strategy was to first compare morphological and physiological characteristics of these congeners to identify potential niche differences, and then explore whether these physiological differences reflected differences in their distribution at some spatial scale. Starting with physiological and morphological characteristics, myself and my crew examined a suite of physiological characteristics at sites in the Baraboo Hills where these two congeners co-occur. And today I'm going to focus on 5 traits in particular. So starting with the top row of this graph, I want to look at specific leaf area, which describes variation in how broad and thick a leaf describes variation from narrow, thick leaves to broad, thin leaves. And all else being equal, plants that have broad, thin leaves will have an advantage in shady environments. And on the right, I'm going to look at light response curves, which describe how photosynthetic rates vary with light availability. And so what you could expect is that species that are adapted to shady conditions might peak earlier and reach peak photosynthesis earlier than plants that are adapted to sunny conditions. When we examined nudiflorum and glutinosum, we found no difference in specific leaf area or in their light response curves. So next we decided to examine 3 additional traits to get a better sense for what they might be doing. So on the bottom row, going left to right, we have the leaf height of flowering plants, and all else being equal, plants that hold their leaves higher are going to be better competitors for light. They can reach light that smaller statured plants can't. In the middle, we have carbon-13 isotopes, and plants that have higher carbon-13 isotopes are plants that more strictly regulate their water use. And then finally on the right, we have leaf carbon to nitrogen ratios. So plants that have high carbon to nitrogen ratios are more nutrient conservative, and they tend to allocate more of their resources to structure and defense. When we looked at these co-occurring Hylodesmum congeners, we found that glutinosum is taller, more strictly regulates its water use, and has higher carbon to nitrogen ratios. From these differences, we would infer that glutinosum should have an advantage in sunnier environments where increased water stress should favor the increased allocation to structural tissue and more stringent water use regulation. So what we decided to do next was to take a look at their distributions and see whether these two species segregated. When you look at their broad-scale distributions across Eastern North America, what you'll see is that their ranges are broadly overlapping in these two figures on the left. But what our expectation was that when we looked at their ecological distributions within the state of Wisconsin, that we'd find these two species would segregate along environmental gradients in light and water stress. However, what we found is that their ecological distributions overlapped broadly. And although glutinosum extended into sunnier sites, they didn't segregate from one another along this gradient. Instead, the distribution of nudiflorum fell entirely within Hylodesmum glutinosum's ecological distribution. Importantly, where these two species co-occurred were in moderately shady forests, in which glutinosum's primary competitive advantages, being taller and being able to more stringently regulate its water use, would be diminished in these moderately shady sites. In other words, where these species are co-occurring is where they're going to be the most equal competitors. This suggests that equalizing processes could promote co-occurrence in those moderately shady microsites where the competitive differences between the species are relatively small. So to obtain a finer scale picture of Hylodesmum distributions and population dynamics within sites where these species co-occurred, I established several study plots within the Baraboo Hills. Within each of these plots, we mapped the distribution of individual plants. And in one of the sites, we mapped the distribution in 2 consecutive years, allowing us to look at local population dynamics over time. So when we looked at static distributions, what we found is that individuals of a given species were spatially clustered, and juveniles were clustered near adults. This is perhaps unsurprising, but it's consistent with the idea that dispersal should be especially important in environments where equalizing processes play an important role. In the site where we mapped populations in consecutive years, we were able to overlay those maps with one another to take a look at fine-scale population dynamics. And what I want to illustrate here is that we found very different demographic properties between glutinosum and nudiflorum. And I'll draw your attention here to that last box, which reflects annual population turnover. And we found that nudiflorum had almost 20% higher annual population turnover than glutinosum. With only 1 year of data, it is difficult to make robust inferences about local population dynamics. However, when we look at these differences in demographic turnover in reference to their physiological characteristics, the 2 points I want to make are first that nudiflorum has increased turnover that results from both increased rates of mortality and increased rates of recruitment. And the second, this is consistent with the idea that there might be a life history trade-off that could promote co-occurrence within these sites. Nudiflorum, in addition to having higher population turnover, less stringently regulates its water use, is an inferior competitor, and allocates less to structure and defense than glutinosum. And so there could be alternative life history strategies where nudiflorum, even though it's a weaker competitor, if its population is cycling faster locally, it could promote co-occurrence among these two species. So wrapping this section up here, physiological characteristics influence Hylodesmum distributions, but there's little evidence of niche segregation. Instead, these congeners are co-occurring in habitats where there are more equal competitors. From this, we see that equalizing processes could play an important role in allowing these species to coexist with one another, or at the very least, setting the stage for demographic differences and life history trade-offs to facilitate coexistence. Next, let's examine how local environmental variation influences plant distributions and coexistence at finer spatial scales. Just as environmental variation between forest communities may influence broad-scale plant distributions, local environmental heterogeneity could also influence finer-scale plant distributions. The primary difference between broad-scale environmental sorting and the role of local environmental heterogeneity is the increased potential for dispersal to scramble species-environment relationships that form at local scales. Thus, the influence of fine-scale environmental heterogeneity on local plant distributions and the potential for coexistence depends critically on the scale of dispersal, the spatial scale of environmental variation, and relative competitive differences between species. To examine the potential for environmental heterogeneity to influence local plant distributions and coexistence, I studied an easily overlooked source of environmental variation in temperate forests: soil depth and the distribution of trees. Subtle variation in soil depth and proximity to trees can create fine-scale variation that has the potential to influence forest herb distributions. For example, shallow soil microsites may be unable to support plants with substantial below-ground organs, and this may favor shallowly rooted plants in those microsites. Similarly, when you have reduced soil volume, there could be reduced nutrients or reduced water holding capacity that may favor plants that are more nutrient conservative and slower growing. One cause of variation in soil depth in temperate forests is the distribution of trees. So if you've ever taken a walk through the forest and looked at the base of a tree, where the roots are coming out, in between those root buttresses are shallow soil microsites, shallow soil pockets that form. So for the reasons listed above, we might expect shallowly rooted plants, more nutrient conservative plants, to be found closer to the base of trees than plants that are further away in deeper soils. But there are other factors that influence the environment, the near environment at the base of trees. For example, after it rains, rain flowing down the stem of trees can lead to increased leaching of soil nutrients immediately at the base of trees, and it can also wash away fine sediments, leading to reduced water holding capacity. As a result, we might expect plants that are relatively nutrient conservative, slow growing, and shorter to be favored at the base of trees. Whereas further from the base of trees on deeper soils where there's increased water availability, increased nutrient availability, we might expect taller statured species with increased nutrient requirements to have a competitive advantage. To test these predictions, I selected 3 focal species. Starting from going from left to right, we have hepatica, bloodroot, and trillium. So hepatica is an annual evergreen that holds its leaves for 1 full year before replacing them. It's relatively short-statured. Its leaves rarely exceed 12 centimeters in height. It has a shallow fibrous root system and relatively low nutrient requirements. In contrast, bloodroot and trillium both complete the majority of their photosynthesis early in summer. They are taller than hepatica, with sanguinaria having an intermediate height and trillium having a taller height. Both species possess a thickened rhizome from which there are deeper and fibrous roots that extend. Bloodroot has intermediate nutrient requirements and trillium has high nutrient requirements. So from these physiological differences, what we'd expect is we would expect hepatica to be restricted to shallow microsites and be more common near the base of trees, whereas bloodroot and trillium should have a competitive advantage on deeper soils with more nutrients and would competitively exclude hepatica. But these 2 species may be unable to establish in shallow soil microsites or near the base of trees. But one important similarity that I just want to note between all 3 of these species is that they all possess lyosomes or oil bodies on the seeds, which are dispersed by ants. And ants are rarely dispersing these seeds more than 1 meter or 2 in horizontal distance. So to test hypotheses about how these species might respond to fine-scale variation in soil depth and the distribution of trees, I established a 50 by 50 meter plot in McGilvra Woods State Natural Area and mapped the distribution of all trees as well as every individual hepatica, bloodroot, and trillium within this plot. And so in this map on the left, you can see an overhead view looking down at the plot, the distribution of hepatica, bloodroot, trillium, and then trees within the plot. When we were mapping the plants, for each plant, we recorded soil depth up to 50 centimeters and using a wire probe, and then we recorded the distance to the nearest tree. And using these data, we can test our predictions about how these species should be distributed at relatively fine spatial scales. So I'm going to show you two graphs. On the left here you're going to see the average soil depth for each of the three focal species after we account for spatial autocorrelation and soil depth. And then similarly on the right, we're going to show you the average distance to the nearest tree for these three focal species. Consistent with our predictions, Hepatica occupied shallower soil microsites than either bloodroot or trillium, were not different from one another. And furthermore, Hepatica was found in closer proximity to trees than either bloodroot or trillium. There was a moderate correlation between soil depth and distance to nearest tree in the plot, but it was relatively weak. So taken together, these findings are consistent with our predictions based on plant morphology and physiology, and in terms of how these species are responding to local environmental variation. So to better understand whether these differential responses to local environmental variation might promote coexistence, we characterize the spatial associations between individuals of the same species and between individuals of different species. So this graph is going to show you the results of that spatial point pattern analysis. The top row is looking at spatial associations between individuals of the same species, and the bottom row is looking at spatial associations between individuals belonging to different species. In our point pattern analysis, what it does is it compares the observed probability of finding a pair of individuals at a given distance, which is that lag distance on the x-axis, to the probability you would expect of finding pairs at that distance if individuals are distributed at random with respect to one another. And so in our graph, that solid line is showing that observed probability divided by what's expected, and the expected probability is shown by that dashed line. And then there's a gray simulation envelope that illustrates the range of possibilities for what we'd expect if plants are distributed at random in space. And so there's kind of a nice little key here. When that solid line falls above the simulation envelope, the individuals are aggregated in space. In other words, there's a higher probability of finding a pair of individuals at that distance. And when that line falls below the simulation envelope, we infer that plants are segregated in space. In other words, that the probability of finding a pair of individuals at that scale is lower than what we'd expect. So starting with looking at spatial associations between individuals of a given species, we found that for all three focal species, individuals were aggregated or clustered within 1 to 2 meters. In contrast, when we looked at individuals belonging to different species, we found that individuals of different species were consistently segregated beyond 2 meters. These findings are consistent with the idea that plants are spatially partitioning microsites within forests. Furthermore, I want to note that the spatial scale of conspecific aggregation, which is 1 to 2 meters, heterospecific segregation, typically beyond 2 meters, match closely with the scale of dispersal, which we infer to be 1 to 2 meters for these antidispersed species, as well as the observed scale of environmental variation in soil depth, which was 1.7 meters in this study. Taken together, these results suggest fine-scale environmental variation can shape local plant distributions within temperate forests. But just as importantly, this local environmental heterogeneity can facilitate coexistence when differential plant responses to environmental variation are coupled with limited dispersal and fine-scale environmental variation that match closely in terms of their spatial scale. So the final process I want to talk about today is interactions between plants and pathogens and their potential to promote coexistence among forest herbs. Plants, like people, must contend with pathogens that may compromise their ability to grow and survive. These pathogens are expected to spread quickly when plant hosts are clustered together in space or occurring at high population densities. Unlike us, however, plants do not have the agency to moderate their exposure to pathogens by social distancing. Consequently, the accumulation of pathogens at high plant densities or in close proximity to host plants are expected to increase plant mortality, especially among seedlings, which are most vulnerable. These Jansen-Connell mechanisms are known to affect plants in many, many ecosystems. But relatively little is known about how these Jansen-Connell effects might affect temperate forest herbs. However, there are tantalizing hints of the potential importance of pathogens. Almost all forest herbs are infected by a host-specific rust fungus, and you can see two examples of this with mayapple on the top and zigzag goldenrod on the bottom. So in this study, I wanted to examine how interactions between plants and soil pathogens might affect herb growth and performance. But more generally, interactions between plants and soil microorganisms can consist of either antagonistic interactions, such as pathogens, or potentially mutualistic interactions that may improve plant growth. And so generally, I was interested in understanding the character and variation in these plant-soil interactions among forest herbs. So I conducted an experiment to estimate plant soil feedbacks in 9 herbaceous plant species pictured here. So going from top left to bottom right, we have white snakeroot, columbine, bottlebrush grass, geranium, pointed leaf tick trefoil, Jacob's ladder, Virginia knotweed with its lovely magenta chevrons, zigzag goldenrod, and early meadow rue. My experiment consisted of growing each focal plant species in soils with and without soil microbes present, and I autoclaved soils to remove soil organisms that were present. In addition to conducting experimental treatment with and without soil microbes, I also manipulated soil origin. So half of the plants were grown in soils that were derived from conspecific individuals or parent plants, and half of the soils were derived from plants that were heterospecific individuals of other species. And so after growing these experimental plants for 3 months, I harvested the plants, dried their tissue, and weighed biomass to estimate plant growth. By comparing plant growth in the different soil treatments, we can evaluate the nature and host specificity of plant-soil interactions. And so I'm going to show you 2 graphs here. On the left, I'm going to show you the ratio of plant biomass production in unpasteurized soils versus pasteurized soils. So soils where those microbes are present and soils where the microbes are absent. So starting with the graph on the left, when a point falls to the left of that dashed line, it indicates that plants grow better when soil biota are absent. And so when we look at that left panel, what we see is there's considerable variation in the strength and direction of plant soil feedbacks among these 9 species. At the top, we have Thalictrum dioicum, which was strongly inhibited by the presence of soil microbes. Whereas at the bottom, when we look at geranium and pointed leaf tick trefoil, we see that this growth of those species was enhanced by the presence of soil microbes. So in addition to just looking at the presence or absence of soil microbes, we can also, if we look within experimental pots where those soil microbes are present, we can compare plant growth in conspecific trained soils versus heterospecific trained soils to understand how whether those interactions are host-specific or more generalist. So in the graph at the right, when a point is negative, it falls to the left of that dashed line, it indicates that plants grow worse in home soil or soil derived from a conspecific individual, suggesting that there are host-specific pathogens that are influencing plant growth. In contrast, when points fall to the right of that line, it indicates that plants are growing better in those home soils. And what you can see looking at the variation among these 9 species is that there's still considerable variation. And one kind of really interesting one that pops out here up at the top is Thalictrum dioicum, which not only has reduced growth when soil microbes are present, but its growth is most inhibited when they're in soil derived from conspecific individuals. And so this is a classic case of Jansen-Connell mechanisms at work. These findings reveal considerable variation in how plant-soil interactions influence the growth of herbaceous plants. And this variation likely has consequences for local plant distributions and the potential for coexistence. For example, the top 5 species, those that are generally inhibited by the presence of soil microorganisms, rarely form monospecific clumps in nature. They often space themselves out and are relatively sparse in their local distributions. In contrast, the bottom 4 species, which exhibit neutral to positive interactions with soil microorganisms, often form dense monospecific clumps in nature, either as the result of clonal growth or as the result of seedlings growing up near adult plants. This experiment suggests that plant-soil feedbacks in forest herbs have the potential to influence plant growth and potentially facilitate coexistence among some species if antagonistic interactions with host-specific pathogens prevent any one species from becoming too common. More generally, interactions between plants and soil biota, including both antagonistic and mutualistic interactions, play an important but previously underappreciated role in temperate forest herbs. So returning to the big picture, the goal of my dissertation research was to evaluate the ecological processes that drive community assembly in herbaceous plant communities in temperate forests. And in doing so, I had hoped to evaluate whether the predictions commonly made by ecologists about which ecological processes influence plant community assembly and the spatial scale at which those processes operate were supported in temperate forest herbs. At relatively broad spatial scales, my investigation of herbaceous plant distributions across the landscape revealed how functional traits influence plant responses to environmental variation. These findings are consistent with common assumptions that trait-environment relationships are largely responsible for shaping broad-scale species distributions. My findings at finer spatial scales within forest communities, however, revealed that a more nuanced suite of factors may influence local herb communities. And in this talk, I evaluated 4 of those potential factors. Contrary to common assumptions about the role of niche differentiation and complementary resource use in structuring local plant communities, we found little evidence that co-occurring species exhibited niche differentiation. In contrast, strong environmental sorting among communities seems to lead to co-occurrence among functionally similar species. And as a result, equalizing processes resulting from relatively small competitive differences and local dispersal may promote co-occurrence among these functionally similar species, or at the very least set the stage for other weaker processes such as life history trade-offs to facilitate local coexistence. We found fine-scale environmental heterogeneity can influence local plant distributions and promote coexistence when plants spatially partition microsites within forest stands. And finally, interactions between forest herbs and soil microorganisms, including both antagonistic and mutualistic interactions, may play an important but previously underappreciated role in structuring local herbaceous plant communities and diversity. Taken together, this research provides insights into the diverse array of ecological processes that structure herbaceous plant communities and temperate forests, as well as the spatial scale at which these different processes operate. These findings improve our understanding of plant community assembly in temperate forests and may have relevance for many other plant communities. Beyond these basic advances, an improved understanding of community assembly in temperate forests and the processes maintaining herbaceous plant diversity will contribute to conserving the spectacular diversity of herbaceous plant species in temperate forests. My PhD research would not have been possible without the support of numerous individuals, organizations, and institutions. Throughout graduate school, I was funded by an NSF Graduate Research Fellowship, and my research was also supported by grants from the UW-Madison Graduate School, the Botany Department, the UW-Madison Arboretum, the Wisconsin Botanical Club, and the Botanical Society of America. Many individuals helped shape and improve my research. I'm grateful for my PhD committee for their advice and support. Don welcomed me into his lab and encouraged me and was flexible as my interests changed over time, and his advice ultimately led to the research that I have today. I thank Tom for sharing countless insights into plant ecology, and you can probably see many of his ideas percolating up through my research. Brett, Monica, and Tony were generous with their time, with their advice, and helped shape my research and continue to provide excellent guidance for me today. Beyond my PhD committee, I'm fortunate to have an array of mentors who have been with me for nearly 10 years now. So huge thanks to Nancy, Mark, Dan, and Stuart, who have so fundamentally shaped my development as a scientist. I also want to thank a number of other individuals, Daijong Lee, Sarah Johnson, and Dave Rogers, in addition to leading efforts to collect the broad-scale plant distributional data that I used earlier in the talk. These individuals have been generous with their time and sharing insights and encouragement over the past 4 years. Similarly, Rick Langkow and Rich Henderson have shared their expertise generously. I also want to thank Kate and Sarah for being kind of my 6th and 7th committee members and always offering excellent advice. Over my time as a graduate student, I've been fortunate to work with a number of fantastic undergraduate students. Brooke, Caitlin, Grace, Riley, and Kevin all helped collect the data that I presented today, and none of this work would have been possible without them. I'm also grateful for the volunteers who helped me collect a number of this data and were not paid for their efforts. And I also want to thank the Givnish Lab and the Damshin Lab for helping me refine this presentation. It was really generous of you. Finally, I'm grateful for the support of family and friends. During my time in grad school, I made a number of friends. Many of these friendships were short-lived, and come to think of it, weren't always terribly mutual, but they nonetheless made a lasting impression. But jokes aside, I'm grateful to all the individuals who have offered support, encouragement, and friendship over the past 4 years. Really special thanks to my lab mates, Allie, Rachel, and Janine. My decision to enroll here in graduate school was largely motivated by my interactions with these individuals, and my time over the past 4 years has not disappointed. These individuals are wonderful scientists and even better people. So thank you to all the family and friends who made this work possible and supported me. At this point, I would be happy to field questions that folks might have.
Thanks very much, Jared. That was wonderful, admirably clear. We do have time for questions now from the general audience, and later when we go into closed sessions from your committee. Who wants to handle those? Allie or Rachel, were you going to do that?
So I can kind of explain. There are 2 ways that we can ask questions here. The first way, at the bottom of your screen, there's a little small person that kind of is raising their hand. And so one option is if you raise your hand, I'll see a little notification pop up, and I can call on you, and then you can, I think you'll be able to unmute yourself and ask a question. Alternatively, there is a chat window, and you can type in a question to the chat window, and I think Allie was going to, she can kind of take care of asking the question virtually for you. So one of those two methods of asking questions should work just fine.
I've got a comment here from Bill Stoneman, who says, "Congratulations, well done. I worked on a wildlife habitat mapping project in Alaska and Minnesota post-graduation from his bachelor's in forest resources using remote sensing and ground truthing. And he can relate a lot to your studies. Good luck."
Thanks,
Lots
Bill.
of other congratulations in here as well. Looks like Tyler has a question.
Did Tyler raise his hand?
Yes.
Tyler, can you unmute yourself? No.
I think you might need to change the permissions, Jared, or he could just type it.
Yeah, if you can type it in, Tyler, that would work.
Yeah, they can't unmute themselves.
While Tyler is typing that, I have a question for you, Jared.
Shoot, Allie.
In your project looking at the local environmental heterogeneity, it seemed like soil depth and the dispersal capacity of these species was really, really important. But you also mentioned when you were talking about the differences between those three species that some of them had variation in nutrient requirements.
Sure.
And some of the work from my current department is looking at differences between, for instance, oaks and maples in terms of stem flow and through fall, and finding maples have a much higher concentration of nutrients right around the tree bowl compared to oaks. And I don't know if that's something you can look at in your data, but I was wondering if that was something that you had considered at all.
That's a great question, Allie, and it's definitely something that I've thought about. Within my study plot, I think it was something like 95% of the trees were sugar maple. So there wasn't enough variation to look at how strongly those patterns of aggregation near tree stems might be affected by tree identity. But I mean, it's certainly something to consider if you get outside of, if that plot was in a forest stand that wasn't predominantly sugar maple, if there weren't like 6 other trees, if you had a mix of oaks and maples, it'd be really interesting to look at how that would provide probably the best example or the best situation outside of an experiment to look at how, or to try to tease out the relative role of physical soil depth versus the actual nutrients that are available.
Great, thank you. Okay, Tyler typed in his question here. It says, "Great and very interesting work. I'm wondering if and how all of these mechanistic insights could be used to inform conservation of rare, endangered, or perhaps common but important species at the scale of the Baraboo Hills, or more broadly."
Yeah, great question, Tyler. I mean, I think that, one of the kind of the motto behind a lot of conservation and restoration is, if you build it, they will come, right? If you create the right habitat conditions and those species can get there, that they're going to persist and survive. And so I think generally that is good advice. And so knowing what are the conditions that these species require, predominantly a lot of managers are thinking broader scale and thinking about at the scale of forests, how do we maintain oaks, for example. But where research like this could be helpful is in thinking about what are the actual mechanisms that are maintaining diversity at local scales. So if it is fine scale environmental heterogeneity, that could inform how conservation practitioners are managing these forests to maintain some level of local environmental heterogeneity. Or if, for example, these sort of interactions between plants and soil microorganisms are really important. Thinking about how to maintain intact soil microbial communities. And so for me, it's those kind of insights that might be relevant to thinking a little bit more critically about, not just creating the broad scale environmental conditions necessary for these plant species to survive, but thinking a little bit more mechanistically and at finer spatial scales about what can be done from a conservation and management perspective to continue to promote diversity in these habitats. So hopefully that answers your question, Tyler.
He says, thanks, that makes sense. Heterogeneity is good. I'm not seeing any other questions just right now, but a lot of congratulations coming through on the chat box and saying how amazing you are and what a good job you did.
Well, thank you everyone for attending. If anyone has questions, my email is up on the screen here, and feel free to shoot me an email if you have any questions.
Thanks again, Jared. And the applause is deafening. You just can't hear it. We're going to adjourn for 5 to 10 minutes. That is your committee, and then we'll recongregate at the other link you sent us at that time. You might want to bring us all in as moderators to make the audio and video sharing easier at that point.
Yeah, I think I have it set up for that one, but I'll make sure that I do. Thanks, Don.
Okay. See you in a minute.
All right. Thank you, everyone.
Bye, Jared. Awesome job. Have fun.
Thanks,
Take care.
Rachel.
We'll talk soon.
Sounds good.
Did you remember to stop recording too, Jared, and save it?
No.